Today we are looking at endomembrane trafficking, which has to do with protein synthesis. Proteins are made and transported through the process by and in different endomembrane organelles. To understand endomembrane trafficking, we need to take a look at these organelles as well as the process of protein synthesis. The nucleus is very important because it stores genetic material called DNA, or deoxyribonucleic acid. The first part of protein synthesis is called transcription, and we will take a closer look at that over here. On transcription, the DNA is in a double helix, so the hydrogen bonds between the two strands are essentially unzipped. Then an enzyme called RNA polymerase binds to this DNA and it creates the complementary mRNA strand seen here. There are 4 nitrogenous bases that will keep your DNA together, called thymine, adenine, guanine, and cytosine. But in this new mRNA strand, instead of having thymine, you now have uracil. And then once you have your mRNA, 3 different bases together is called a codon. And this is very important to keep in mind. Now, this all happens in the nucleus, but once it's created, it leaves through a nuclear pore and either goes to a free-floating ribosome in the cytoplasm or a ribosome on the rough endoplasmic reticulum. So at this point, it's going to go through translation, which we will take a closer look at over here. Translation is the part where actual protein synthesis takes place. Each of those mRNA codons are codes for an amino acid, which are what proteins are composed of. Basically, this molecule thing here is a tRNA, and a tRNA holds an anticodon, which is the 3 complementary codons, or the 3 complementary bases to your codon. Each tRNA molecule has a specific amino acid, and it brings all of these amino acids together, and it creates this polypeptide chain, and this polypeptide chain is your actual protein. So once this polypeptide chain is complete, it undergoes what's called post-translational modification. This is where it's folded into a 3-dimensional shape in the endoplasmic reticulum before it continues on. That would happen over here, but obviously we can't see it. So now it goes through this entire endoplasmic reticulum and it gets to the end. Now the protein needs to get to the Golgi apparatus, a cellular organelle that will package and ship the protein to where it needs to go. To do so, a section of the ER will bud off containing the folded protein. This section that buds off is called the transport vesicle. The transport vesicle must dock on the Golgi apparatus. The location where the vesicle docks is called the cis face, which is the loading side of the Golgi. The material in the vesicle is then dumped into the cisternae. The cisternae are flat chambers on the Golgi where modifications and packaging take place in the Golgi. The protein continues on through the cisternae until it reaches what is known as the transface. This is where the Golgi ships or sends off the finished product. This occurs similarly to how it did in the ER. A vesicle buds off with the complete protein in its vesicle, and there are 2 different types of vesicles. One type will become a lysozyme, which is necessary, or which remains in the cell and has digestive enzymes to break down unwanted material. The other type of vesicle is called a secretory vesicle, which is necessary in exocytosis. Exocytosis is the process in which a secretory vesicle will fuse with the plasma membrane of the cell, and the vesicle will end up outside of the cell. The plasma membrane is a phospholipid bilayer that surrounds the cell. It is a semi-permeable, meaning it allows certain things in and out of the cell, and other things must remain outside or inside of the cell. So basically this would go through, bud off, fuse here, which you can see, and then it would let off your protein outside if necessary, or it would remain in the cell. Overall, this process displays endomembrane trafficking and the role of the endomembrane organelles in transportation of proteins. Thank you.